All right, welcome to our lab 8 on two-way anovas. So let's remember that two-way anova, though we haven't calculated them by hand, what they deal with is more than one nominal variable or category that anyone can belong to. All right, so our research situation here is looking at Parkinson's disease, which is a neurological disorder, which has an array of symptoms, of the main one being tremors, numbness, but it's long been thought that estrogen contributes to the occurrence and symptom expression of Parkinson's disease. So we'll be looking at data from a study investigating the effects of estrogen-based hormone treatments on alleviating symptoms of Parkinson's disease. So we're going to take a look at it. We want to see how the drug, where the treatment compares to the placebo or lack of treatment, and we're also interested across gender because we have a prediction here that treatment might lower symptoms in females but increase symptoms in males due to the fact that we have different estrogen in our bodies. Right? So that's our prediction. And those are our different sets of categorical variables. We're going to hop over into SPSS. We're going to start by putting these variables into variable view. So let's go ahead and do that. So we have our 4 variables that we're going to input here. We're going to have a participant number. We're going to have gender. We're going to have estrogen, whether they receive the treatment or not. And then we're going to have the Parkinson's symptoms. So let's go ahead and hop over into SPSS and do that. So we have participant. We have, what comes next? We have participant, gender, estrogen, and then we have the Parkinson's symptoms. All right, so we have some codes that we want to use. Code gender 0 and 1 for male and female. That's not what we wanted. 0 and 1 for males and females, and then we also want, for estrogen, we want 0 for placebo and 1 for treatment. So let's go ahead and get those values in here. So 0 male, hopefully we're getting a little more used to this here. Again, it's just to simplify our data without losing meaning. So when we're working with very small data sets like this, it's often not a big deal. However, it's good practice, or if you're ever working with a very large data set, to have things kind of simplified in this way. Okay, so we got placebo and treatment, okay, and of course we want to change our measures here. So if we hop back over, participant is not anything that we'll be calculating with, so it's not important, but we do want to indicate whether we have categories. So our gender and estrogen are going to be categories, and our Parkinson's symptoms will be on a scale. Right? Then you're going to input the data below, and I have... so here we are with our headings. I went ahead and put this in earlier, so I have it here so we can work through, but you will be putting this in yourself here. Then we're going to run an ANOVA, but we do it a slightly different way, and we use something called the general linear model. So let me go ahead and hop over, and we're going to analyze general linear model. And then we only have one option here, univariate. So the general linear model is just meant to be a very flexible tool in which we can use multiple types of data and multiple variables at once in order to analyze group differences. So it asks for fixed factors, which is what the ANOVA in our previous lab asked for. So this is where you're going to put your categorical variables, and then our dependent variable is Parkinson's symptoms. That's our ratio dependent variable. So if I scroll down here, you can see this here. And we could also have what we call covariance or random factors. This is sort of the next step up in statistics. It's, say, we also want to control for age. Say we also want to control for education level. There are other controls we could basically put into an analysis. And we could do that here, but we're not going to do that here. We're just going to stick to a basic two-way ANOVA. But we are going to go into plot. And we're going to create a plot where we're plotting our 2 groups, the estrogen and gender groups. So let me go ahead and do that. We go in here, we'll put gender on the horizontal axis, and we'll put the estrogen treatment on the separate lines. So here we have that. We also want to go into options and display our means. So we're going to have them list a few things here. So under options, move all of our variables over to the display means for box, and then a few things we want to click into. So let's go into, okay, we're okay there. Okay. Does it not want to let us add? There we go. Again, always just ahead of myself. Okay, so options here. I want to do descriptives, effect size here. All right, continue. And then we have 4 options here. We also our estimated marginal means. So let's go ahead and do that as well. Okay, let's go ahead and get done here. Our estimated means. I also want to pull in this gender by estrogen, as well as these two here. So that's what we have here. And continue. So these are in separate ones here as opposed to this previous version of SPSS, which is okay, but there we have it. All right, so then we're just going to go ahead and run this. We're basically running it all at once. Might take a minute to turn it out since we have a bit going on, but let's see what it gives us. So we can see here our different breakdowns of what we have. So we have gender and our placebo treatments. And then here we have descriptive statistics where we have right? So a number of individuals that belong to both of these groups. So we have males that receive the placebo, the treatment that receive the placebo, and females that receive the treatment. So that's the basic idea behind a 2A ANOVA. A factorial design, we call it, where we have combinations of our nominal variables, and then here we have a big long output, right? Notice how many things we have here, but what we're looking for is the relationship between gender and estrogen. So we would look here to this significance value, and notice that it is significant, which is a good news if you're this researcher, right? Finding a significant effect. It gives us some of our descriptive statistics here, mean, standard error, as well as confidence intervals, separated plots here, gender and estrogen. And then we have a sort of plot that shows the interaction between the two groups. So we have the placebo group and the treatment group. We have gender points here. And then, of course, this is representing our Parkinson's symptoms. So you can see this sort of interaction, right? One group sees improvement, and the other sees a sort of, we can just compare groups, right? So females over here who received the treatment, males over here who received the treatment, females here that received the placebo, and males here that received the placebo. So we can start to take a look at the interaction, the differences across our groups. So yeah, so that's it for this lab. You do want to include your output that has the analysis output, the descriptive statistics, the estimated means, and the means plot, and you will, of course, be saving and submitting this output or .sppv file. So please be in touch if you have any questions, and good luck.